Okay, welcome everyone. Welcome to our webinar. For those of you who don't know me, my name is Charlene. I look after the Entomological Society of Southern Africa's media and marketing. So we are super privileged today to have Catherine Sol with us. Professor Catherine Sol is based at the University of Pretoria. Catherine and I actually found a photo of us during our undergrads together on a field trip. So I've known Catherine actually a long time. She's currently a professor at the Department of Zoology and Entomology in the Faculty of Natural and Agricultural Sciences, and has given us kindly her time today to present amazing work. She's known for working on many invertebrate species, especially dung beetles and lacewings. And thanks, Catherine. We really look forward to hearing all about your research. So there'll be an opportunity afterwards for questions. So yeah, over to you, Catherine. Thank you.
Great. Thanks, Charlene, and welcome everybody. Yes, I know, I still remember our undergrad together. Yeah, we had lots of fun. It takes me back, but hopefully I'll show you guys some photos that also take me back. So I'm going to get right into it. Oh, I'm hoping that this is going to turn now. Oh, there we go. So I wanted to start this a little bit back to front and start off with the acknowledgements, because what I'm about to present would not be possible without all the students and the collaborators and the colleagues that are very much inextricably involved in my research. So going back to the late 90s, early 2000s, Clark Scholz and Amanda Bastos, who supervised my PhD, and they were really instrumental in guiding me into where I am today. Amanda, as one of the few women in the department, has mentored me a lot over the last number of years, and I really appreciate her support. Clark, with his just also mentorship and support and his general insect knowledge, has gotten me where I am today. And then Mervyn Mansell, a lot of you probably know Mervyn. Both Mervyn and Clark are retired, but Mervyn has also, we've worked extensively on lacewings together, but he's also got a huge amount of general insect knowledge and has guided me and mentored me over the last 20 odd years of my career. Robin Crew, I started off doing an honours with Robin Crew, and I shifted from bees to beetles. And Robin has also been a massive influence in my life as a mentor. He's often given me lots of career advice, which has been really important for me. During my PhD, Sean Forgy, Claudia Medina, during my start of my master's and then my PhD, Sean Forgy and Claudia were around. So was Vasily Grebenikov, all of whom I still have contact with. We spent many hours in the field collecting beetles and having fun and enjoying the time and the work of the PhDs and the MScs together. Then students such as Johan that did an MSc with me, Azimo, I co-supervised his PhD. Christian Descott is doing a PhD with me at the moment. Ishtiaq, I'll present some of her work. She also did a PhD with me. Wagner, who I co-supervised his PhD. He's now at the Ditsong Museum. I have a lot of contact with many of these people because we're still working together. Vinicius is a postdoc that's just joined my lab, and it's really great to have him around. He's been here for almost 2 years, and we're hoping to extend a little bit. Agatha Morelli, who worked on urban biodiversity with me. Yusuf is my go-to as a collaborator for looking at CFCs. So this is cuticular hydrocarbons, CHCs, sorry, cuticular hydrocarbons. So insect communication. He worked extensively with him on that. And then to Ian Engelbrecht and Shannon and Almay. Shannon, I'm going to present some of her work on trapdoor spiders. She did honours, masters, and PhD. And Almay did an honours and a master's with me on wrinkles, but I'm not going to talk about her work. It's completely a little bit off topic, but we spent lots of time in the field. To the more recent people that I've started working with, Nigel Barker and Paulette Bloomer on the Waterberg project. We looked at biodiversity in the Waterberg. Kathy Bester is the postdoc that's joined us on the project. And then to the even more recent collaborators, Alan Ellis, John Midgley, Genevieve Turan, and Kurt. We're looking at pollinating Diptera. We've got some nice money to look at flies and pollination. And then to Isarita, who did her PhD at the same time that I did. She's a friend and a colleague Mike Bruford, who passed away 2 years ago, and Paulette and Eileen is a PhD student. We're working on a massive project together, which we've dubbed Framework GD. And we're looking at genetic diversity at the moment, just across the savanna biome. So we've sampled dung beetles. We will look up the west coast of Southern Africa at a later stage, but it is a massive project. And then last but not least, to my husband, I think a lot of you know Christian Perk. He has supported me unconditionally over the last 16 years. He's been an advocate for me in every step of the way. And I can't thank him enough. And then to our gorgeous little miracle son, Finn, who has just turned 5. And all of you know some of the long hours we worked, and he is so patient and understanding with me. And for that, I cannot thank him enough. And then Chris Weldon, I'm not going to talk about our work, but Chris and I have worked together for the last 5 years, mainly on thrips in avocado and macadamia. But we've also we're looking at expanding that to working on berries and possibly table grapes. And Rashid from NWU, he collaborated with us on Johan's project on dung beetle duck microbiomes. So just to acknowledge all these people that have been instrumental in what I'm going to present going forward. So without further ado, I wanted to start off with Pachysoma. So this is Pachysoma glentonii, and it's commonly referred to as it's commonly referred to as the galloping dung beetle. I'm not sure why I'm getting echoes here. Can everybody hear me?
Yes, we can hear you, Catherine. Can everyone just make sure
Okay,
their
great.
microphones
Can you still
on?
hear me?
Yes.
Okay, great. Sorry. So just to jump into Pachysoma, it's a flightless genus of dung beetles. It consists of 13 species. They've got a very interesting set of feeding and foraging adaptations, which I will chat about just now when I get to the slide. They're endemic to the arid areas along the west coast of South Africa and Namibia. They have really interesting biogeography, which I will talk about. And this was the first molecular phylogeny ever of the genus, and it was one of the first molecular phylogenies generated on beetles in Africa, not the first, but one of the first. And this is a group that is of huge conservation concern due to habitat threats. So for those of you that know the Western and Northern Cape, you've got large-scale wheat farming in the areas. You've got lots of commercial development. You have mining for diamonds and other minerals. And then what came to light a lot during my PhD is the collection and resale of the genus. There were a lot of illegal collectors that were very interested in the group and in what I was doing. And at some stages, they would offer me you could get for up to $5,000 for one of the larger beetles on the black market. The same goes for colophon. This is Colophon promosi. Colophon is one of those beetles that is a collector's dream, and you can get up to $15,000 for one of these on the black market, which makes my heart very sore. So going down memory lane, Charlene, this is some of our first field trips. At that stage, we didn't have cell phones or cell phone cameras, and no, we didn't have a digital camera, so this was all taken on film. And this is in Sossusvlei, where we did some work in Sossusvlei. This is Gobabeb, these two photos here, Gobabeb. This is just before we crossed the Kwiesep River into the dunes to collect. Yes, there's a hand.
Can we please save all questions for after the talk, please? Thank you.
Great, thanks. Yeah, so Claudia took a lot of these photos on the film camera that she had, and we scanned them in. You can see Mervyn, Claudia, Clark, and myself, and this is one of the assistants that helped us in Gobabeb to actually collect. But it's just nice to know, go down memory lane and see all these photos. So to get back to Pachysoma and its foraging and feeding habits, so Pachysoma it feeds on dry dung pellets and detritus, and the beetles actually comb the food into their hind legs with the fur or the hairs on the back. I don't know if you saw the hairs, not the fur, on the back of their legs, and they drag it. So they drag the food forward. They then dig a burrow like this to below the moisture line, and they will bury the food below the moisture line, and the moisture reconstitutes the microbes associated with the food, and then they can actually feed on the food. So it's actually quite, it's actually really, I could spend hours watching them, as you can see here from the video footage. So to get right into it, I generated a molecular phylogeny for Pachysoma, looking at the 13 species. All of them except for 2 were well supported. If you have a look up here, you can see that there was a complex. I called it the Hippocrates-Glentonii complex. And this wasn't well resolved, but it was just one individual. And I never got back to investigate it, but I think adding data and or individuals would actually clarify the species complex. All the other species were very well supported. I actually got, it was really nice because this was one of the first phylogenies that we started to date. When we started to do dating, we dated it, and I used mutation rates because I didn't have fossil data available for Pachysoma. So that seems like the genus diverged in about 3 million years ago. And then what was really interesting when we looked at this lineage here was the oldest lineage at about 2.66 million years ago. And all the species that were in this or are in this lineage, so Hippocrates, Glentonii, Aesculapius, and Androidii, all occur in the southern part of the distribution of Pachysoma. And this lineage here, lineage 3, was the youngest of all the lineages, and it dated back to 2.4 million years ago. And it, interestingly enough, has all the species that occur right in the north. So this is Denticolus, Rotundigenus, and Rodriguezia, and they have the most northerly distribution of Pachysoma. And then these species, though you have this rapid radiation, and this rapid radiation of species occurred between 200 and 600,000 years ago, which is very recent. And this got us to thinking, why would you have the oldest species in the southernmost distribution and the youngest species in the most northern distribution? And we went back and had a look at the biogeographical history of the area. And we know that the Namib Desert has a high level of endemic fauna and flora. This is very well known and very well documented. But when you go back and you read the literature, you will see that there's 2 schools of thought about the fauna occurring in the desert. And the one is that you've got ancient, that the fauna is ancient, it's really, really old. So you have these really slow rates of evolution dating back to the Cretaceous, which is about 66 to 145 million years ago. But then you also have younger, faster rates of evolution. So you have these 2 so-called contrasting ideas where you have the younger, faster rates of evolution, and these would date back to diversification going back to between 5 and 23 million years ago, which was during the Miocene. Now we know that the hyperaridity of the Namib from geological evidence dates back to about 15 million years ago. So we see that we have rapid radiation in Pachysoma. I mean, it dates back to just under 5 years. And the question that we actually wanted to have a look at is what has driven this rapid radiation? And there were 2 things that we could actually see or find. The first one was if we look at the Barken dunes. Now, what are Barken dunes? If you go and have a look at aerial photos of the desert, you will see that these are these alluvial dunes, and they look like this. They're not permanent dunes. They're alluvial sands that originate at the tertiary river mouths. So what do I mean by tertiary river mouths? Go back. That would be things like here you've got, I can't see, you've got the Olifants River, the Buffels, the Khoikhab are very good rivers for these. And what happens is these sands accumulate at the river mouths and they're blown in a northerly direction. We know from work by Android Younger, when he looked at satellite images, that these Barken dunes move in a northerly direction and they can move anywhere between 10 and 100 meters per year. We also know when we go back and we have a look at different species, so different taxa. Android Younger saw this for tenebrionids. Lorenzo Prendini documented it in scorpions. But we know that speciation events for these arid adapted taxa are occurring in and around the riverbeds. And you can see it distinctly here. Orange River, you've got Beniximi, you've got Fitzsimonsi here at the Khoikhab. So you've got this, and Aesculapius further down. You've got these very distinct speciation events that are occurring at the rivers on these alluvial sands that are being northerly blown, blown in a northerly direction. And Androida Younger coined the term pocket speciation. And what he actually meant by this is that these dunes are blown with the species in a northerly direction. Some of the dunes get left behind at the riverbeds, and then you get speciation occurring in those areas. And then the rest of the dune may be blown in a further northerly direction. And in that way, you get this pocket speciation occurring at these riverbeds, and then the taxa can then move out as the area allows for them. The second thing that we looked at was water availability. So for those of you that have visited the desert, you may know that you get the formation of this fog. And it's very common along the west coast, along the arid adapted areas. And we know that the fog dates back, we can date it back to about 1.8 million years ago. So how does that fog actually form? Up the west coast of southern Africa, you have the cold Benguela Current. So there's cold air above the Benguela Current, and then above the land, you've got the warm air. And when the cold and the warm air come into contact with each other, you get condensation occurring, and then you get the formation of this fog. And this fog can be blown up to 50 kilometers inland. There's often a wind that blows across from the south to the northeast of the country, or across South Africa and Namibia, and it blows that fog inland. And what happens when that fog comes into contact, for example, with plants or animals, condensation occurs, and in that way there is free water available for them, for those that actually require water. We know from Mitchell et al., this paper was published in 2020, that you can get up to 25 days per year of fog. And if we look at the fog basking tenebrionids, they're very well documented. They sit here on the top of the dunes, on the peak of the dunes here, with their abdomens facing upwards. And as that fog is blown inland, you get condensation occurring, and the water then runs down into their mouth, and they have the availability of free water. And we hypothesized that this would also have affected Pachysoma diversification. So there were two things, the bark and dunes and the availability of free water from the fog, that would have affected Pachysoma diversification. I'm going to jump a little bit and change taxa. I'm going to talk a little bit about lacewings. I'm assuming, because I'm talking to entomologists, you all know the order Neuroptera, but I can never resist a good photograph. So the ones that are often well known, most people know the antlions. I'm not going to talk about antlions, specifically the pit building antlions, of which none of these are pit builders, but I just wanted to show you the adults, and you can see the larvae that are hugely predaceous. I'm going to dwell more on the family Nemopterinae, the subfamily Nemopterinae, but the family Nemopteridae is characterized by these beautiful extended hind wings on the adults, and they've got these paddles. And you've got two subfamilies, the crocines, which are called threadwings, and then the nemopterines, this is a nemopterine. They've got these spoon or ribbon wings, and you can see the spoons on the hind wings. The hypothesis behind the spoons is that it's used for stability during flight. If you ever watch them fly, they drag their hind wings behind them as they move. So it looks like it has a stabilizing effect, and then also for camouflage. Some of their hind wings are lichen colored, and when they land on a rock or on the sand, they actually disappear. You can't see them very well. They're very well camouflaged. And there's a third hypothesis, which is that they're used for thermoregulation. There are two contrasting views. There is support for it, and in some other groups, there's lack of support. So this is something that can be further investigated and could be quite interesting. The adults, as you can see here, quite distinctly, have got these elongated mouth parts, and they are pollen feeders. And they are good pollinators on the groups or on the plant species that they actually feed on. All the larvae of this group are predaceous. You can find them in small caves, hollow tree trunks. Some of them you just find them in the sand. You get litter dwellers, and then you get those that are inquilines found in the nests of ants. So just to show you, I mean, look at those paddles. They're absolutely beautiful. And you can see if that just flattens on a rock, you get that idea of camouflage. So this is the other subfamily, the crocines. You can see they've got these thread-like hind wings, The reason for this is they are mostly found in caves or rocky overhangs, and these thread hindwings are used as a sensory function to sense the edge of the cave. They also have quite an elaborate mating, if I can call it, dance, and they use the hindwings for that as well. The larvae of the crocodilians are quite different. They're really cool. They've got these extended necks, and the neck and the head is sclerotized. The thought behind this is that because they're predaceous, but if you watch the crocodile larvae, they actually run over the top of the sand. They don't normally bury themselves in the sand. I think that's just due to the habitat, the rocky overhang or the caves that they found in. If they catch an ant, as I'm sure most of you know, ants bite, and if they turn around and they bite the larvae, they will bite on the head, the sclerotized head or the neck, and that is so they're protected, so the larvae are protected. Because if the abdomen is punctured, then the organism will die because it's under hydrostatic pressure. Interestingly enough, arbitrary fact of information, these were first discovered in the tombs of Giza in Egypt. They were thought to be funny cockroaches, but when they eventually bred them out, they realized that they were Neuroptera. Just a little cool story to add. So why this group? And I'll explain to you the values in brackets now, but what I'm presenting now is only based on the values not in brackets. So the family Nemopteridae, there's 142 species worldwide. 43 are crocines, 99 are nemopterines. 72 of those occur in southern Africa, and 57 of those are endemic to the Western and Northern Cape provinces. So there's huge. We have a lot of endemic taxa in South Africa, in Southern Africa. The subfamily Nemopterinae, there were 11 genera. 62 species found in Southern Africa. 51 of them are endemic to the Western and Northern Cape provinces. So if the group, so as I said, there's lots of endemic taxa in Southern Africa. Just to give you an idea of the distribution, so you can see up the west coast and into the desert. You've got many of the genera and many of the species. We also see them in the Nama Karoo as well as in the savanna, but not as diverse. So what Mervyn and I wanted to do is we wanted to look at the validity of the 11 genera. And as you can see, I generated 4 gene regions, and I'm just presenting a Bayesian phylogram here because it's easy. But basically, the red dots represent maximum likelihood, parsimony, and posterior probability support. And you can see that Nemia, Barbibuca, Holterina, Knash, Lactea, Palmipena, and Dorinkia are all well-supported genera. Chasmoptera is also well-supported, but it's highlighted in green. It's really interesting. This is an Australian genus with 3 species in it. And it's really interesting from a biogeographic point of view that it pops up here. And this is something that needs further investigation. So the genera Nemopterella, you can see, is paraphyletic, and Nemura, Sisioptera, and Semirinkia need a little bit more work and further investigation. And then there's a new genus and species. Based on this study, and I just want to highlight this because I want to highlight the diversity that we discovered. One of my PhD students, Ishtiaq, she revised the genera Nemopterella and Nemia, and she basically added data and then reanalyzed my data, and it was really interesting. So Nemia, and the reason why she revised these 2 genera was because the morphological characters to separate them were not well documented. So we wanted her to specifically have a look at the morphology, but it was even bigger than we actually thought. So what she ended up doing was she ended up splitting Nemopterella into 3 genera. So you've got Nemopterella sensu strictu, which sits here. We've only got 2 species. We couldn't get the 3rd species for molecular work with Africana and Cedrus. And then she described Afrotera, which is the most derived genus sitting here. It has 28 species in it, of which 8 were newly described in her PhD, which is quite a mean feat in my mind. And then you've got the genus Secanda, and this is a monotypic genus. So what I wanted to highlight here, and I'm not going to dwell on her work, is just how much undescribed diversity we discovered just through looking at 2 genera. But to jump back to the previous study, I had this really nice phylogeny that was well supported. So what I could do was I could use the dating methods to date it. And there's really good fossil data for the Nemopterines or for Neuroptera in general. And I could actually, the two stars, I could put fossil dating on the phylogeny, and I could then go and date it. The subfamily is old, but we know this. It dates back to about 145 million years ago, but we know this from fossil data. So that wasn't a surprise. We just supported what we know from fossil evidence. But what was really interesting is that there were two things that I could see with the dating. You had this slow accumulation of taxa here, which you can see by this green block and these long lines. And this dated back to the Middle Eocene, Middle Miocene, so between 44 to 11 million years ago. And then more recently, you had this recent rapid diversification, which occurred in the last 4.5, 5 million years. So going back to what I spoke about in Pachyssoma, where you had these two schools of thought, where you've got ancient and recent, you're now seeing these two schools of thought overlaid in one subfamily. And the question that we wanted to ask was, why are we seeing this? And when we went back and had a look, if you have a look at the slow accumulation, if you have a look at the time period 44 to 11 million years ago, there was a gradual change between semi-arid to arid, and it was interspersed with wetter periods. And we hypothesized that this allowed for this gradual accumulation of these taxa, where you got these long branches. And then more recently, you have this rapid diversification, and we hypothesized that this was due to the radiation of the flora. So we know from unpublished work I think it was a master's student, that some of the species feed on the Azoceae and the Asteraceae. I'm not even going to try and pronounce this. And we know that their radiations date back to 3.8 to 7.8 million years ago, which links really nicely with the Nomopterine radiation. And we know that you had glacial and interglacial cycles during this time period. So we hypothesized that during the time period you would have the expansion and the contraction of the flora, and that the species associated with of Nemopterines that were associated with the flora would expand and contract and radiate with the flora. So that was really nice, showing that you had these two patterns and that there were two slightly different drivers driving the slow accumulation of taxa and then the rapid diversification. I'm going to change taxa completely and jump to, it's still stick with invertebrates, but I'm going to talk a little bit about baboon spiders. And the reason why I wanted to talk about this is because this has direct conservation implications. And Ian Engelbrecht approached me, and we started the study, and what we wanted to look at was the validity of the genera of the baboon spiders in Southern Africa. So just a little bit of history. I don't know how familiar you are with baboon spiders, infraorder Megalomorphae, and then you get the purseweb spiders, which are the Atypidae. You get your typical trapdoor spiders, which are your ctenizids, your megids, and your idiopids. We'll talk about the ctenizids and the stasimopids just now. But then you get your baboon spiders and relatives. So you've got your certocenids, your nemesids, and your dipleurids, and the theraphosids. And I'm going to dwell a little bit on the subfamily Theraphosinae, so the theraphosids. This was just a rule of thumb that Ian put together for baboon spider identification. So it's just a broad way of identifying baboon spiders. So you can look at the scopulae, which are patches of short bristles on the sides of the fangs, and you can see them here very distinctly in Pteronoculus. So the two genera, Harpactierella and Brachinopus, don't have bristles on the fangs. Harpactierra has bristles here. You can see them on the outside and the inside of the fangs. Ceratogyrus has the outside of the fangs only, but Ceratogyrus is the typical horn, has got the typical horn that you can see here in this photo and in this one here. It's a beautiful photo. Oops, sorry. And then Orgocephalus has also got a scopulae on the outside only, but it's supposed to be a big orange spider. And then Idiothele on the outside only again, but if you flip it over, it's black underside. Pteronoculus, here you can see distinctly on the outside only. And if you flip Pteronoculus over and you look at the underside, it's got a brown or gray underside. When we go, I can't resist, sorry. So this is thanks to Taki. These are just beautiful. I mean, look at those colors. Beautiful photos. So when you start to look at the thorax of the spiders, you can see here, you can just have a look at the colors. You can just see how diverse they are. So we assumed just from looking at these photos that you had a much higher diversity than we had originally thought in the baboon spiders. I generated, I think, 4 or 5 gene. Oh, wait, so hold on. So baboon spiders are protected under the Transvaal Provincial Nature Conservation Ordinance that was in 1987. And what they did was they classified that, well, they put 3 genera, which were supposed to be protected from wild harvesting and trade. This was Ceratogyrus, Harpactira, and Pteronoculus. They were considered commercially threatened and therefore were listed on the IUCN Red Data list, and that was on account of their popularity as pets. And then they are listed on TOPS, and this is also due to the fact that they need to be managed in an ecological and sustainable manner, but also due to their popularity as pets. But then what happened in the last 10 years or so, the taxonomists started to revise a lot of these genera, which is perfectly fine, but they started to shift species from one genus to another genus, and this rendered a lot of species without protection. So what Ian and I wanted to know was, do we have really good genera, and can we take this to conservation authorities? So I generated 5 gene regions worth of data, and this is just a Bayesian phylogram to show you the support. This is where we got our samples from. You can just link the colors to the names of the genera here on the side. And you can see that we got samples from all over Southern Africa. Fortunately, there's a very large group of people interested in baboon spiders and baboon spider conservation. So they were able to glean samples from us and send them to us. So you can see that Brachionopus is quite a well-supported genus. This lineage needs a little bit of further work. Harpactira overdaki. Pteronoculus looks like a good genus. Ceratogyrus, Orgocephalus, Idiothele. And then at the top here, when we start to look at Tricognatella, Harpactira, and Harpactira, we need a little bit more work here. I think that adding samples and possibly gene regions would help us tease apart where we see these genera that are intermixed. And I think this species has been incorrectly identified, which is why it is actually sitting there. But what this study highlighted to all of us is that baboon spider diversity is completely underestimated, and we desperately need additional sampling. I know that Ian and colleagues are actually doing this on an ad hoc basis. And this is where I would like to bring in the idea of citizen science. I think we underestimate the potential or the possibilities with citizen science. Ian and them run the Baboon Spider Atlas, and anybody can contribute. And in this way, they have expanded the distributions of some of the genera and the species and all these kinds of things just through the assistance of citizen science. And I think this is something that we can draw on. I'm going to jump again, and I'm going to talk also about spiders, but this is very close to my heart. It's looking at the family Stasimopidae, but specifically the genus Stasimopus, and these are trapdoor spiders. This is a beautiful photo, but it's not a South African species. It's a Corsican trapdoor spider. And this is work done by a PhD student of mine, Shannon Brunt. She's now doing a postdoc in France, and then Robin Lyle from the ARC. So this project stemmed from the Karoo Biogaps project, which I think a lot of you may be familiar with. It was a consortium, it was funded by the FBIP, and it was a consortium of over 20 institutions, and we sampled across 14 taxonomic groups. You can read all of them there. And we obviously linked in with the spiders. The Karoo, prior to the start of this project, was one of the most undersampled or studied regions in South Africa. The theory behind this was that due to the harsh conditions, it's quite challenging sampling in the area, and the perception that it was species poor. We did, however, at that stage know that the karoo responds very poorly to disturbances and that it has a slow recovery rate. You can see this by the farmers and the overgrazing and that occurs in the area, and that the ecosystems were sensitive. We also knew that what had been sampled, there were high levels of endemism, but what we wanted to do with the Karoo Biogaps project was basically fill biodiversity information gaps, so add to the biodiversity information on the Karoo. And the long-term goal of this was to assist government policymakers, which would allow them to make informed decisions on land use change. For those of you that know the Karoo area, you will know that land use in the area consists of competing interests such as mining, farming, renewable energy. There was extensive shale gas exploration at that stage, and then you've got the SKA, which is a square kilometre array, all competing interests for land within the Karoo. So we sampled the Greater Karoo area, but we were specifically mandated to sample the fracking area, or the area that was designated for fracking that was under shale gas exploration. And you can see here Nama Karoo and Succulent Karoo biomes that we sampled across. You can see the provinces that we sampled across. What's really interesting is just the purple dots are the localities where we found Stasimopus, and the green triangles are the type localities. So you can see there are a lot of type localities for species that are sitting either directly in this area or just outside of the designated fracking area. We sampled 79 sites. We found Stasimopus at 55 of those sites. It's not to say at the balance at the others there isn't Stasimopus, it's just we didn't collect them. We did 2 years worth of sampling. We did sites, we did, depending upon how close or far we were, we did 3 to 4 sites per day. And the sites were normally about 50 kilometers apart. It was incredibly intense. For those of you that have ever sampled these kinds of ground-dwelling spiders, you've got to literally scrape off because you don't always see the trapdoor, so you've actually got to scrape off the top of the soil, and if you see the trapdoor, then you can open the trapdoor and you've got to dig the spider out as you dig down. So labor intensive, but it was actually great fun. We thoroughly enjoyed ourselves. The Stasimopus is commonly referred to as the corklet trapdoor spider. So this is what the trapdoor looks like when it's closed. And then when it opens, you can see the silk-lined tunnel that goes down. But what I really wanted to highlight, this comes from Engelbrecht, Ian, and Lorenzo Prendini. They published in 2012 on this. This is the female, and that's the male. And they are, so you can just see morphologically, they look completely different. There are instances where these have been described as two species. Now we know that the males look like this. And before the males mature, everything looks like this. So if you dig it out of the ground, and you generally find this. If you collect above the ground, you actually find the males. So morphologically quite difficult to identify. So why ground-dwelling spiders? We know it's an ancient group. They date back to the Carboniferous, which is about 300 million years ago. But despite this time elapsed, they have retained their plesiomorphic characters and ancestral character traits. They have very conserved morphologies, and they're very difficult to identify morphologically. They also have interesting life history traits that make them ideal candidates for biodiversity monitoring. They're long-lived. There's an example of an idiopod in Australia living up to 43 years. It was monitored every year. They went back to it. They live in burrows or chambers, which are lined with silk. The females lead a sedentary lifestyle. They only exit the burrow. They'll jump out of the burrow to catch a prey item and they go back in. The males are nomadic. So these males are the wanderers to find females, and they mate with the females, and they are therefore the drivers of dispersal. Spiders are top invertebrate predators in terrestrial ecosystems. They're also a food source for many animals such as birds, scorpions, and lizards, and they can be important in the life cycle of parasites such as flies and bees. So just a little bit of history. So the male will wander, he finds the female, he mates with her. The spiderlings will then hatch and they live with the females for a short period of time. And then they move out of her tunnel or her burrow and they go and make their own tunnel or burrow a couple of meters away from her. So you can see this very significant structure, genetic structure, that could develop. So Shannon generated data, genetic data, and she drew this phylogeny, and she had 11 completely distinct clades, which took us a little bit by surprise. Clade K, for those of you that are wondering, is from UKZN. So Stasimopus is actually found all over the place. And this individual was given to us, so we included it. So there were 10 clades that were from the Karoo, with 10 possible species. Most of them were completely unique, especially things like in the Tankwa Karoo. And for those of you that have ever worked in the Tankwa Karoo, there's lots of unique fauna and flora. It's very different, that area. So we had, as I said, 10 possible new species to describe. Shannon, unfortunately, there's really good fossil data for megalomorphs, and she was able to do some really nice dating. And what was really interesting for me, and we didn't expect this, is the genus originated during the Paleocene, which is not unusual, approximately 63 million years ago. But what took us by surprise was you had, again, the slow and then this rapid diversification. So the initial clades, so these initial long branches, diversified between 45 and 11 million years ago, which we saw it in the Neuroptera and in the Nemopterines. And then you had this recent rapid, this recent rapid diversification, which occurred from about 5 to about 120,000 years ago. So we were seeing in a completely different group, we saw something very similar biogeographically. And the question was why? So just to let you know, we described 9 new species in the fracking area, just in that Greater Karoo area. There were originally 7. So we took that number to 16 species in the Greater Karoo. That was based on morphology and molecules. So we used both. So what do we hypothesize has driven the diversification of Stasimopus? If you go back and you look at the biogeographic history, in that Greater Karoo area, they've got what they term the Great Divide. And this is where you have large shifts in climate, and it's actually increasing in temperatures in Southern Africa. And because this is an arid adapted taxa, we hypothesize that would have started that slow accumulation of taxa over a number of years. We see this in the idiopids and the halo, I'm not even going to try and pronounce that, in Australia. We see exactly the same trend in Australia in these arid adapted ground dwelling spiders. And then in the last 5 million years, you've had lots of increase in temperatures and very high acidification. And then you've got these glacial and interglacial cycles. And that would have driven the rapid speciation or diversification of Stasimopus. And it mirrors Western Australia and the establishment of the arid zone. And it's really interesting that we see these in two. You see it in Southern Africa, and you also see it in Australia. So again, the diversity of trapdoor spiders is completely underestimated in the Karoo. And the BioGaps project resulted in major diversity being discovered in the Karoo. And the use of the land for shale gas exploration was denied. I'm throwing this out there. I don't have an answer, but I just wanted us to think about it. So the aridification of the Karoo is increasing. We know that from a lot of the guys that are working there on birds and these kinds of things. And we know it's due to climate change and habitat degradation. So my question or my thought was, will this continue to drive Stasimopus diversity, or will the conditions become too harsh? And that will result in the extinction of these arid adapted taxa. So I wanted to pull everything together. So I've spoken about 4 very different taxa with 4 very different life history traits. And one of the things that was highlighted to me going through all of these and working with these different taxa is that conservation authorities use species lists for conservation because it resonates with the public and policymakers. And the fact that we have got so much undescribed diversity makes me think back to conservation and how well we can actually conserve our invertebrate taxa. The other thing that as I've come across extensively is the shortage of taxonomic information and skills. And I am not a taxonomist, but I work extensively with taxonomists. And I think this is a skill set that we need to look at encouraging. We need to highlight the value of taxonomy and our collections. I think our collections are underutilized. We also need to look at social media. How can we use social media responsibly? And how can we bring funding into taxonomy and the skill set? We also need to think about collaboration between conservation biologists, and I've used that as a very broad term just for academics, us as academics. We need to link in with taxonomists and legislators and managers like the Stasi Mopus study or the Biogaps project. I think this is quite important. And the last thing that I wanted to share with you is that what's really interesting is you've got many taxa, but just invertebrate taxa that have these very different, very similar patterns that we're seeing in diversification, but there's very different drivers. And we see this, I mean, there's work on chameleons, there's work on flies and all sorts of things that show these similar patterns. And it's really interesting. I don't know, Charlene, do you want to stop there? Because I can go on, but we can stop.
I think we can do with 10 minutes of questions. Do you can finish off in a
yeah, sorry, I turned my volume off. Sorry, I didn't hear you.
I said you could, if you want to finish up, you can finish up, then we can have 10 minutes of questions.
Okay, let's go quickly. So
Okay.
okay. So I just wanted to go through some of the future studies that we've done. We started to look at gut microbiomes on dung beetles. I don't know how many of you know, but dung beetles actually don't all feed on dung. So your ancestral state is fungus feeders. You've got those associated with rock hyrax. Some of them we don't know. You've got this group here that are mixed dung and detritus and millipede feeders. And then here you've got a lot of your mostly dung feeders. I thought my idea behind this was if I take representative subset of samples across this phylogeny and we look at gut microbiome contents, we can actually plot feeding across the 9 tribes, or there are 9 here, but there's more now, of dung beetles in Southern Africa. Johan did just that. We selected detritus feeders in Pachysoma, carrion feeders in Anaculcus, that's Coptorina. Sorry, I can't see my screen. We've got Scaliages, we've got Digidonthophagus, and then we've got Dicranocara. Thinking that if we had representative samples, that we would get something really strong out of this. So this is what the gut microbiomes looked like. It was absolutely chaotic. And when we started to sort, so we removed the bacteria that we couldn't identify, we got a little bit of a better picture. But even if you have a look here, you can see your 2 millipede feeders in Scaliages brittani and Scaliages hepies. You can see how different their gut microbiomes are. And these are 2 species collected in a similar area. So this opens up the question for what is actually going on here. We want to make this more specific, so drop it down to 1 genus, like just looking at Scaliages. We're not sure if this difference is due to things like natural gifts given by mothers, or if it's linked to feeding on different species of millipedes, or if it's linked to different environments. So these are all things that we need to go and investigate. Talking about biodiversity, I couldn't resist this photo from Henny. We look at an organism and you look at it as a beetle or a spider or a human or whatever it is. But when you flip this beetle over, I mean, you've got these macrochelid mites as well as the thoracic fly on one individual, one dung beetle individual. So I think biodiversity is far more than what we actually often think about. If you just go and look at your microbiome, your gut microbiome. The next step is looking at this idea of comparative biogeography or phylogeography. This is an up-and-coming PhD, which is funded by the DGD through the African Museum, which is based in Teferi in Belgium. The project is called the Dip Project. And what we want to start looking at, because I haven't presented any of this, is we have lots of data. Macroderies is the dung beetle genus that occurs at the west coast. I didn't talk about Afrotera, but we've got the 3 genera, Nemopterella, Mimi, and Afrotera. Again, we have really nice data. This is from Genevieve Turon, a publication of hers. She's got some nice data on Prosecco. I don't know if I'm pronouncing it correctly, Jen, you can correct me if I'm wrong, but you can see this rapid diversification. Megapulpus, so this is Alan Ellis. Again, you can have a look and you can see this beautiful distribution, and then you've got these this rapid diversification. And then lastly, this was unpublished at the time that I set up this presentation. I'm not sure if Cassie has published, but she might have. Look at this beautiful up the West Coast. Start to overlay these taxa with different life history traits and have a look at what are the fine scale drivers of what the diversity that we're actually seeing. So these are some of the questions we want to answer. We are expanding on our urban biodiversity, but we're going to focus on pollinating flies. So we showed really nicely in a study in 2019 with Agatha and a landscape architect called Ida, where if you plant native plants in an urban environment, you have increase in diversity. Now what we want to do is we want to expand that, and we want to look at only pollinating flies, but we want to resample Gauteng, so resample the Future Africa Native Garden. We want to sample in Pietermaritzburg and then in Cape Town and do a comparative study with these native gardens that you find, and then your typical garden that somebody plants where you've got these strelitzias and these kinds of things in the garden, and compare biodiversity across. And the idea behind this, all of you know that we have a declining insect population, but the idea behind this is that we can look at urban greening, encouraging people to plant plants that attract insects, which I think is really good. I'm working with Venetius and Wagner. We're looking at trogons. So similar to what I've shown you previously, understudied diversity, and we want to start looking at the biogeography. So that's the other thing we're looking at. And then I didn't mention this, and I'm not going to dwell on this too much, but we sampled, and I just want to show you about biodiversity. We sampled the Waterberg across 6 elevational sites. And 56,000 specimens were sorted. So Wagner and Drake sorted 56,000 specimens, 130 species, many of them undescribed. So just think about the species richness and abundance. We're going to barcode some of the well-known species, describe some of the undescribed. Previously, Kruger was thought to be the most diverse with 100 species of dung beetles. The Waterberg has sort of, changed this thought process a little bit. None of this could have been done without my funders, but I just want to dwell on Henny. All of the photos that you've seen, or most of the photos that you've seen, were taken by Henny de Klerk. He's a phenomenal photographer of insects. I couldn't have done this without my collaborators and students that I mentioned in the beginning. And then all the funders, the FBIP, we funded the Waterberg project and the Crew Biogaps. Stanby, who funded us through the FBIP for our baboon spider work. The ARC with Robin, who helped us with fieldwork for Stasimopus. The NRF has been very supportive of me with rated and unrated funding. And then the DGD through the Africa Museum in Tervuren. We've just got nice funding through them for the Dippo Dip project. And then because I cannot resist insect photos, thank you, and thank you for listening.
Thank you. Thank you very much, Catherine, for that absolutely inspiring talk. Congratulations on all your amazing work and helping to increase South Africa's insect diversity. So great stuff. We have some time for a few questions. If you could raise your hand, or you could see here, there's one from Gimo.
I can't hear who's asking questions. Can you hear me? Sorry, I can't.
Yeah, I can hear you.
Okay.
Gimo, you can go ahead.
Hi, Gimo. I can't hear you.
No, me neither. I think he's muted. Any other questions? The floor. While we wait, yeah, Catherine, I just want to ask you, what is the way forward even for Colembala? We found exactly the same pattern, this huge diversification in the same time period. So what is the way forward that you can maybe think of that one can accumulate all this data to maybe see what's the underlying drivers? Are some the same, or some taxa different?
Yeah, that's what we hope. I'm hoping that we're going to get the PhD student that we're looking at. Well, we don't have anybody, it'll be advertised, but I'm hoping that we can answer that for where we have really good. So like Alan, for example, they're actually sampling, the flies, and they're linking the species to the plants, and these kinds of things. So I'm hoping that with that fine scale data, we're going to be able to start teasing apart these patterns. I didn't present it. We've just published a paper on Afroterra where we actually looked at their diversification, because we've got really fine scale data, and we think that's also linked very strongly to plants and the winter rainfall regime, for example, along the west coast. But teasing apart is going to be difficult. But I think the more data we accumulate, I mean, I didn't know that you'd seen the same time periods in Kalembala, and I find it really interesting. I know you see it for, I can't think of her name now. She's published on the chameleons. Lorenzo Prindini saw it in scorpions. It's just unbelievable that we get this very distinct pattern. So I think, I don't know. For me, it's my passion. I love it. It's where it's sort of like where my interest lies. And I'm hoping that with some of the fine scale studies, we can start teasing it apart. I don't know if you guys have started to do that yet or not.
Yeah, no, not really. I think a lot of it boils down to maybe what they eat, So I think, like you said, the basic biology and taxonomy of many taxa are unknown, and that's sort of a first step. I
Yeah,
think just knowing what do they eat, like the lacewings, for example,
yeah.
don't really know exactly what they eat, the adults at least.
And they're pollen feeders. So we
Yeah.
know that, we know what they feed on. There's the gut
Okay.
content analysis, so that we can link quite nicely to the
Okay,
plants.
great.
Yeah.
Fantastic. Any other questions? You could raise your hand or type in the chat or Q&A. Don't see any at the moment. I think I will maybe leave it there. I can just mention in the chat or in the Q&A, there's a form that was posted by our ECR representative, Ashley. If you could fill that in. If you're interested to give a webinar, you can just email me or Ashley. And again, thank you so much, Catherine, for your time. And again, congratulations. It was really nice. If I could ask everyone to just quickly, Catherine, you can stop sharing your screen and just put your cameras on. I just want to take a quick fun photo if people will allow me to. Just put your cameras on for like a second and then I can take a photo. We had almost 80 people in the room, so we're
Oh,
very,
wow.
yeah, Catherine, you're a crowd puller. Okay, great. Thanks. Yeah.
Thank you, everybody, for being there. I appreciate it.
Yeah, it was fantastic. Okay. Hold it there.
Oh my goodness, Vaughan's got a whole room full of people there.
Yeah, that's fantastic. So there were more people. I've got to take a few photos because there's like 4. Hi, Tom. Tom from Cardiff is here. Great stuff. Okay, last one.
Ah, Tom, hi. I didn't even see you.
Cool. I think I got you all. Okay, thanks everyone. Have a lovely afternoon. And I'm sure if you want to ask Catherine more questions, you can find her online at it as well. Great stuff. Thanks, Kath. Have a good afternoon.
Thanks, you too. Bye.
Bye-bye.